Okay, now it's time to go through the SAM file format in much greater detail. As stated previously, SAM files consist of two parts, the comments and the alignment data. When it comes to the comments section, there's not much more to say here. As stated previously, these comments can be used to record notes and various metadata, just as they can be completely absent. Different alignment software and different SAM file processing tools each use their own system when recording metadata via these comments. We don't need to know what these systems are in order to understand the core pipeline, so there isn't much point going any further. When it comes to the alignment data table, however, there is a lot more to talk about. As previously stated, different rows are separated by new line characters, while different cells of the same row are separated by tabs. Each line represents an alignment of a read against the reference genome, but this is not necessarily a one-to-one relationship. One read in the FASTQ file can produce multiple rows in the results, i.e., multiple alignments. This is rarely done because the resulting SAM files could be enormous. Nevertheless, it is possible to specify that the alignment program do so. Let's talk about the table columns in a bit more detail. The first column, read ID, is fairly simple. It should be the same as the ID of the corresponding read in the input FASTQ file. The second column, the bitflags column, is probably the most convoluted and difficult to understand. To properly understand the bitflag system, we would need to understand basic computer architecture. In computer science, a bit is the smallest possible unit of information. It can be either 1 or 0. Typically, 1 represents true/yes, while 0 represents false/no. The information for multiple flags can be stored as consecutive bits. For example, 4 flags with the values of yes, no, no, yes would be 1, 0, 0, 1. If read as a binary number, this would be equal to the decimal number 9. In reverse, we could break the number 9 down into 8 plus 0 plus 0 plus 1, and reverse engineer the bit flags from that number. Theoretically, this allows us to store a lot of information with just a number which can save space, but in reality, this is stupid. It saves very little hard drive space, while at the same time, it is virtually the opposite of easy to read for humans. Using a single unique character for each active flag would only increase the memory footprint of a SAM file by a few percent, if that, and be much easier to read and interpret. But the established format is what it is, so we have to just deal with it. Now, before I explain what each of the flags are, I will need to explain what I mean by the word template. When I talk about SAM files in bioinformatics, a template refers to a biological sequence of relevance which consists of an individual read or the fragment implied by a read pair. In paired end sequencing, a template is a fragment which was sequenced and consists of two reads, located at the start and end of the template. The next read in template would thus mean the other read in the pair. In single-end sequencing, each read can be considered a template by itself, which makes the term somewhat redundant in that context. Now, let's talk about the bit flags and what they mean if set to true. The first flag indicates that this alignment was performed on a read which is part of a template which contains multiple reads. The second flag indicates that all reads in the template, which this read is a part of, map to the reference genome and in a sensible way. The third flag indicates that this read is unmapped. The fourth flag indicates that the next read in this template is unmapped. The fifth flag indicates that this alignment is against the reverse complement of the specified position in the reference genome. The sixth flag indicates that the next read in this template is mapped against the complement of the specified position in the reference genome. The seventh flag indicates that this is the first read in the template. For this pipeline, it would mean that this is the first read in the pair. The eighth flag indicates that this is the last read in the template. For this pipeline, it would mean that this is the second read in the pair. The ninth flag indicates that this alignment is not the best alignment for this read. It should only appear when users allow reads to yield more than one alignment result each during the alignment process. The 10th flag indicates that the read for this alignment fails platform or vendor quality checks. The 11th flag indicates that the read for this alignment was determined to be a PCR or optical duplicate, also known as sequencing noise. The 12th flag indicates that the read obtained via sequencing was split up into two or more subsequences, and this is the alignment result for a non-primary subsequence. And that should cover it. The third and fourth columns denote the genomic coordinates of the alignment. The third column contains the name of the chromosome which the read aligned to. The names in this column will be identical to the names used to generate the reference genome index. If the read did not align to anything, then this column will contain an asterisk. Meanwhile, the fourth column contains the chromosomal positional index of the alignment. The underlying concept itself is quite simple. Every nucleotide in a chromosome has a positional index. The very first nucleotide in a chromosome is base pair number 1. The second nucleotide is base pair number 2, and so on. The index number works like the coordinates of a one-dimensional map. When a read aligns to a part of a chromosome, it will align to more than one nucleotide, but there is no need to record the indexes of all the nucleotides. Only the leftmost positional index is recorded in the fourth column. Numerically, this will be the index with the lowest value, while biochemically, this will denote the nucleotide closest to the 5' end of the chromosomal DNA strand. It is very important to note here that data processed using different genome assemblies of the same organism cannot be combined. This is because the same DNA sequence can be given different positional indexes in different versions of that organism's genome. For a quick heuristic glance, data processed using one genome assembly can have its genomic coordinates converted to the approximate equivalent in a different genome assembly, using software such as Liftover. However, the reliability and accuracy of this translation is not guaranteed. For stringent statistical analysis, data should always be run through the same pipeline under the same conditions. The fifth column contains the mapping quality score, also known as the MapQ score. The MapQ score is calculated based on the probability of the alignment being incorrect. MapQ equals -10 times log p, where p is the probability that the alignment is incorrect, while log is in base 10. Long story short, the higher the MapQ score, the more reliable the alignment is. Keep in mind that the MapQ score is a comparative statistic. If multiple locations in the reference genome tie for the best alignment, then the MapQ score is 0, because we have absolutely no confidence in either of the alignments. Therefore, a high MapQ score does not just indicate good base pair matching, it also indicates a lack of competing alignment site candidates. The sixth column contains the compact, idiosyncratic, gapped alignment report, also known as the cigar string. The cigar string is a number of letters and numbers which report how a query sequence, in our case, the read, aligned against the reference sequence, in our case, the reference genome. A cigar string will contain one or more elements. Each element consists of a number and a letter. The letter, known as the operator, indicates the alignment result, while the number indicates the number of nucleotides. On this slide, I have described several cigar operators and what they mean. For those of you who are not familiar with database terminology, query sequence here refers to the sequenced read, while reference sequence refers to the chromosomal sequence. At least in the context of this current slide, that is what those terms mean. An M is used to denote nucleotides being paired up, whether that pairing is a match or a mismatch. A D denotes a deletion or gap in the query sequence. An I denotes insertion or a gap in the reference sequence. An equal sign denotes matching bases, while an X denotes mismatching bases. H stands for hard clipping, while S stands for soft clipping. They function like deletions, but at the ends of the query sequence. According to the official SAM file specifications, hard clipping should be used when a read is chopped up into subsequences which align to different parts of the genome and are recorded in separate entries. The chopped off nucleotides will not be present in column 10 of the current SAM entry, and will instead be present in a different SAM entry. This prevents the same nucleotide sequence from duplicating as a result of being present in multiple SAM entries. When the clipped nucleotides are not used in a different SAM entry, then soft clipping should be used. At least, this is how hard clipping and soft clipping should be used, according to the official SAM specifications, but some programs do things a little bit differently. Back to the commonly found cigar operators, P stands for padding and denotes a gap which is in both the query and the reference sequence. This is used for multi-query alignments. Finally, we have N, which indicates query nucleotides which align past the end of the reference sequence. It works like clipping, but for the reference sequence instead of the query sequence. This occurs when the reference sequence ends, but the aligned query sequence keeps going, such as when the read aligns past the telomeres. These are the main ones. There may be more operators out there, but they won't be covered here. On this slide, we have several examples. The main thing to note is how 6M can be used to denote 6 aligned nucleotides, whether they are all perfect matches, like in the first example, or whether they contain some mismatches, like in the second example. The 7th column and 8th columns function like the 3rd and 4th, in that they contain a set of genomic coordinates following the same set of rules. However, these coordinates are for the alignment results of the other read in the read pair. For single-end sequencing, where there are no read pairs, the 7th column will always contain an asterisk. Similarly, the 8th column will always contain a 0 for single-end sequencing data. The 9th column contains the size of the fragment that was sequenced. While this can be calculated from the other available information, it is convenient to have as a readily available data value, because we cannot obtain the size of a fragment by simply subtracting the index of one read from the other, as you can see from this figure. The 10th column contains the biological sequence, which is often, but not always, identical to the sequence in the corresponding entry in the FASTQ file. Similarly, the 11th column contains the quality scores of the sequence in column 10, and this is often, but not always, identical to the scores of the corresponding entry in the FASTQ file. The first 11 columns are standard across all SAM files, but more columns may be added which contain additional information. Different bioinformatics programs may put different information into these additional columns, and there is no standardized system for what information is included, nor how that information is formatted. I won't go through all of them, but I will give an overview of the one used by Bowtie, an alignment tool I am fairly familiar with. Under this system, each piece of information recorded consists of three parts: the tag, the type, and the value, separated by colons. The tag consists of 2 characters and specifies the bioinformatic nature of the information being stored. Examples of this include the alignment score of this alignment, how much weaker the next best alignment is, and so on. The type consists of a single letter and specifies the format or type of data. Examples of this include integer, string, char, and so on. Lastly, the value is the data value itself. For example, the tag AS signifies the alignment score, which is stored as a number. The type I indicates that the value will be an integer, so ASI10 records that the alignment has an alignment score of 10. Multiple such pieces of information are typically separated by white space characters. Keep in mind that this is just one of many systems for denoting additional information in a SAM file, and the one you come across in your project may be different. And that concludes the segment on SAM files.